Okay, so this lecture is going to be divided into three parts. First part going to be about the structure of a nerve. Second part, how a nerve impulse is generated. And the third part about all the different supporting cells that help the nerves do their job. Now this is a picture of a pretty typical nerve cell. This is called a multipolar neuron. I've got a separate slide describing the different kinds of neurons a little later on, but this one has a pretty characteristic feature. It's a good one to use for models. Now, this slide, I want to focus on the part up here, the cell body. Okay, the cell body has a substance inside it called Nissl substance. Now, as we go through the rest of this course, as we start studying different systems, you're going to find cells have different names for the same structures. For example, Nissl substance is the neuron's name for rough ER, rough endoplasmic reticulum. Remember, the function of the ER is to transport materials, and if it's rough, it's going to be transporting proteins that were made, because ribosomes dot across the surface of rough ER, and ribosomes make proteins. So this is all inside the cell body, making the stuff that you're going to need through all parts of the cell. Now, in the other cells, our other cell lecture, we talked about the cytoskeleton. Well, the nerve cell has neurofibrils, which is basically the nerve cell's cytoskeleton. It's a bunch of fibers, cytoskeletal fibers. In particular, they're called intermediate fibers that are going to give support and shape to the nerve cell. So it's just not a wiggling blob of mass. Nerve cells will, of course, have a nucleus. No cell can live without a nucleus. And just like before, it's the control center. And within the nucleus, most nerve cells are going to have a very, very large nucleolus. The nucleolus is the organelle that actually makes the ribosomes. And ribosomes are going to be deposited on top of the Nissl substance. So it all has to work together. Okay. Now, this slide I set up here to focus on the extensions that you see coming off the cell body. There's two varieties here. First one I'm going to talk about are the dendrites. In my picture, and here's why I picked the multipolar neuron for this example, dendrites are very numerous. Dendrites on this particular one are all these little tiny extensions coming off the cell body. There's many dendrites of a multipolar neuron, and their job is to carry an impulse toward the cell body. I can't overemphasize that. Dendrites carry information toward the cell body. In my multipolar neuron that I've got here in this picture, you'll have one axon. Now, if dendrites carry an impulse or message toward the cell body, axons carry the message away from the cell body. So when a nerve carries an impulse, you can imagine this, it's going to start out here in a dendrite. It's going to be carried along the dendrite to the cell body. It's going to be carried to this region here called the axon hillock before it passes off to one long axon. That axon might be about 3 feet long. We're talking about a nerve that's going from your spinal cord all the way down to your big toe. So they can be quite long. Now, at the axons will end in what we call an axonal terminal. This is an enlargement over here of the end of an axon. The axon right here bulges out into the axonal terminal. This axonal terminal is filled up with little tiny vesicles that contain neurotransmitters. Those vesicles that have the neurotransmitter are actually made in the cell body, and they're carried down the axon to their end point, those axonal terminals. Now, those axonal terminals do not touch the next nerve. That's a tough concept for some people to visualize, but when two nerve cells come up to each other, they do not form a connection, a strictly physical connection. Instead, they are separated by a gap. That gap is called the synaptic cleft. So this space right here that's separating the axon of the presynaptic neuron, presynaptic right here, postsynaptic down here, that gap that separates them is the synaptic cleft. And all it is just a small space. The actual whole unit here we call a synapse. So in that previous lecture about the reflexes, we said there's polysynaptic reflexes. Well, those would be reflexes that have several synapses, connections or communication points between two neurons. Now, the way it works is this. When a nerve impulse comes down the axon, follow my pointer here, as the impulse comes down here to the end of the axon, it's going to trigger these vesicles with the neurotransmitter to be released. The neurotransmitter goes into this gap right in here and binds to the next nerve cell. When they bind, they transfer the impulse to the next nerve cell. That's why I always tell my classes it is a chemoelectrical stimulus. Yes, it is involving a change in ions, cross back and forth, but it's also involving chemicals. Now, why do we have this difference? Well, if you were looking for a painkiller, a good painkiller would be one that would block right here. A really good painkiller could block all these receptors on the next neuron, and as the neurotransmitter comes across, if they're blocked, they cannot transmit the nerve impulse. A lot of good toothache drops, that's how they work. They don't block the release of the neurotransmitter, but they block the reception of the neurotransmitter on the next neuron. So the impulse stops. You feel no more pain. Whereas some illegal drugs like cocaine does the opposite. What happens with cocaine is in this gap here, the neurotransmitter that's released in the brain that cocaine deals with is called dopamine. Dopamine will travel across here and bind. Now, normally, dopamine is reabsorbed back into this axon and recycled. Cocaine prevents the release or the reuptake. So therefore, nerve impulses continue to fire, and people experience that high because the dopamine is not being reabsorbed. Now, if you notice all those pictures, the axon was covered with a whitey material. That is called the myelin sheath. Now, the myelin sheath is made up of individual cells called Schwann cells. The way they work is this. Schwann cells will attach themselves to the axon and coil around and around like a jelly donut. over and over and over again until they're nicely, tightly packed around there. And the nucleus gets shoved to the outside, like in this picture right here. The nucleus of the Schwann cells pushed out. Now, Schwann cells just attach themselves to little tiny pieces of the axon. They are separated from each other by a little gap right here between the two Schwann cells. That is called the node of Ranvier. Now, in the next lecture, we're going to be looking at how a nerve impulse is first of all generated and then transferred across the cell. As a rule, and this is a good rule to remember, myelinated fibers will always carry an impulse faster than non-myelinated. Okay, so if you can imagine a cell, an axon here that was lacking all these Schwann cells, that would be unmyelinated. And if we were to race two nerve impulses across the same length, the one with the myelin coating would win the race every time. Now, up till now, I've been using an example of a multipolar neuron right up here. Multipolar. Lots of dendrites, one cell body, one axon, all the way out. Remember, multipolar neurons are the most numerous neurons in your body, mostly because they're the ones that are going to make up most of the cerebral cortex. Most of your brain is made up of these kinds of cells. Bipolar neurons, like right here, a bipolar neuron has one dendrite, one axon, cell body right in the middle. In the body, these types of nerves are very, very rare. You're only going to find them in your eyes and your ears, or like in your retina for your eye, sense of hearing. So basically special senses where you're going to see bipolar neurons. Unipolar neurons, what happens is this. You have the cell body, and it's attached by a very short process to a long axon. The central process is going to go towards the spinal cord. The peripheral process will go out to the rest of the body. You will find these kinds of neurons in the dorsal root ganglion of the spinal cord. Remember, the dorsal root ganglion is carrying sensory nerves. So as a general rule, sensory nerves, like those carrying information about pain, touch, taste, going to the spinal cord, are going to be unipolar neurons. So they're not very rare, but they're not nearly as numerous as multipolar.